Immature B cells that bind self-antigens undergo further receptor rearrangement or die or are inactivated. Once an immature B cell expresses immunoglobulin M on its surface immunoglobulin M, its fate is guided by the nature of the signals it receives through its antigen receptor. This was first demonstrated by experiments in which antigen receptors on immature B cells were experimentally stimulated in vivo using anti-mu chain antibodies. The outcome was elimination of the immature B cells. More recent experiments using mice expressing B cell receptor transgenes have confirmed these earlier findings, but have also shown that immediate elimination is not the only possible outcome of binding to a self-antigen. Instead, there are 4 possible fates for self-reactive immature B cells, depending on the nature of the ligand to which they are capable of binding. These fates are cell death by apoptosis, the production of a new receptor by receptor editing, the induction of a permanent state of unresponsiveness to antigen, and ignorance. An ignorant cell is defined as one that has affinity for a self-antigen but that does not sense the self-antigen, either because it is sequestered, is in low concentration, or does not cross-link the B cell receptor. Apoptosis and elimination of immature self-reactive B cells seems to predominate when the interacting self-antigen is multivalent. For example, the multiple copies of an MHC molecule on a cell surface. This antigen-induced loss of cells from the B cell population is known as clonal deletion. The effect of encounter with a multivalent antigen was tested in mice transgenic for both chains of an immunoglobulin specific for H2KB MHC class I molecules. In such mice, nearly all the B cells that develop bear the anti-MHC immunoglobulin as surface immunoglobulin M, because the presence of the already rearranged transgenes inhibits the rearrangement of the endogenous immunoglobulin genes. If the transgenic mouse does not express H2KB, normal numbers of B cells develop, all bearing transgene-encoded anti-H2KB receptors. However, in mice expressing both H2KB and the immunoglobulin transgenes, B cell development is blocked. Normal numbers of pre-B cells and immature B cells are found, but B cells expressing the anti-H2KB immunoglobulin as surface immunoglobulin M never mature to populate the spleen and lymph nodes. Instead, most of these immature B cells die in the bone marrow by apoptosis. This is because the anti-H2KB immunoglobulin on the immature B cells interacts strongly with the H2KB molecules on the bone marrow stromal cells. And that's immature B cells that bind self-antigens, undergo further receptor rearrangement, or die, or are inactivated. Have a great day.